We are preparing for our Next GEM Single Cell ATAC experiment. The Chromium Next GEM Single Cell ATAC Library Kit v1.1 is stored at -20 degrees Celsius and contains ATAC Buffer B, ATAC Enzyme, 20x Nuclei Buffer, Barcoding Reagent B, Barcoding Enzyme, siPCR Primer B, Reducing Agent B, Amp Mix, and Cleanup Buffer. The Chromium Next GEM Single Cell ATAC Gel Bead Kit v1.1 is stored at -80 degrees Celsius. The Dynabeads My1 silane are stored at 4 degrees Celsius. The Chromium Next GEM Chip H Single Cell ATAC Kit v1.1 is stored at ambient temperature and contains partitioning oil, recovery agent, Chromium Next GEM Chip H and gaskets. Finally, we have our single index plate N, set A, which is stored at -20 degrees Celsius. We have all of our Next GEM Single Cell ATAC reagent kits ready. Next, we will proceed to the transposition step.